To the second technical session of the Basic and Applied Science track of General Sir John Kotalawala Defence University's 15th International Research Conference. It is with great honor and pleasure that I introduce the chairperson of the Chemical Science 2 session, Emeritus, Professor A. M. Abeysekera. He obtained his PhD Organic Chemistry, Queen's University of Belfast, 1977, and later joined the University of Wijayarathna as a lecturer in 1979. Retired as Senior Professor of Chemistry in 2015, having served 2 terms as Head of the Department of Chemistry and 2 terms as the Dean of the Faculty of Applied Sciences. He has been a postdoctoral fellow at the Queen's University of Belfast and the University of Utrecht. Emeritus Professor Abeysekera's main focus of research is synthetic and physical organic chemistry, standardization and biological activity of herbal drugs, with over 45 publications in referred journals and over 65 communications at scientific conferences. He has served as Director of Bandaranaike Memorial Ayurveda Research Institute. Emeritus Professor Abeysekera has also served on the Council of National Academy of Sciences, Institute of Chemistry, and Sri Lanka Association for the Advancement of Science. He was President of Section E2 of Sri Lanka Association for Advancement of Science and a member of Research Advisory Board of National Science Foundation. He is also recipient of GRC Research Award in 1994 and has been honored with DSE by the University of Wijayarathna. Ladies and gentlemen, please welcome Emeritus Professor A. M. Abeysekera.
Thank you for your introduction. We can start the session now. I'm very pleased to be here to listen to the presentations that have been lined up. We have 6 presentations with 15 minutes allocated for each presentation. There will be a bell going at the 10th minute to apprise the speaker that it's time to start thinking about the end. At the 12th minute, you are supposed to end your presentation, leaving 3 minutes for a small discussion. There will be some time allocated right at the end of the session if anybody has any further questions to raise. So we are under strict time management here, particularly being the KDU, so I ask the speakers to stick to their times allocated to them. I would now ask the first speaker who will talk to us about investigation of wound healing ability and chemical constituents of Wathupalu, Mikania cordata leaf extract. This paper will be presented by A.U. Hewageegana. I have got that correct, I hope.
Day. I'm very grateful for this opportunity, and I'm Ayuma Hewageegana from University of Colombo. And today the topic of my presentation is investigation of wound healing ability and chemical constituents of Mikania cordata, Wathupalu leaf extracts, and the paper ID is 253, and other authors are mentioned below in the slide. So Mikania cordata Wathupalu, as you all know, it is an invasive vine which is herbaceous and belongs to family Asteraceae, and that is traditionally used the leaves are traditionally used to cure snake bites, stop bleeding from cuts and wounds, and jaundice and septic sore. And also the leaf infusions and the decoctions are for cold, influenza, and fever, and also bronchitis in children. So M. cordata is rich in vitamin A, B, and also in C, which works against fish poisoning. So here are the objectives of our study. The first one was to evaluate the wound healing ability of the plant leaf extracts, and the second one was to determine the chemical constituents in M. cordata leaves responsible for wound healing. So here is the strategy of the study. Using scratch assay, we evaluated the effect of the plant leaf extract on directional in vitro cell migration. Yes. Yeah, so scratch assay is a common method for evaluating scratch assay is a common method for evaluating in vitro wound healing, which means it is one of the most promising methods for in vitro cell migration and also proliferation. So here what we have done is we created a confluent cell monolayer, and then we made an artificial gap or a scratch, and then we added the particular plant extract there, and we observed a scratch edges movement until the scratch is closed using the ImageJ software. So this is the methodology we followed. So fresh M. cordata leaves were picked, air dried, and we cut them into tiny pieces, and we divided them into two parts, and one was crushed using mortar and pestle, and then we just made two extracts using ethanol and DCM, and the other part was sonicated with two different solvents, that is also from ethanol and DCM. So then we performed the scratch assay only using the crushed ethanolic leaf extracts and with the C6 glioma cells. Then we performed TLC and column chromatography for all the crushed and sonicated extracts. So C6 glioma cells, that's the cell line we used. It's a tumorous cell line developed in brain glial cells in adult brown rats. So in order to perform this scratch assay, we just culture the cells in dishes, and we made a scratch in order to have a wound, and then we incubated the cells in ethanolic leaf extracts in different concentrations like 10, 100, 500 and 1000 microgram per milliliter concentrations, and then we just observed the wound healing using a fluorescence microscope, which is also known as the cell imaging. And then the width of the scratch was measured, and it is analyzed in different time intervals at 0th, 6th, 12th and 24th hour. So from here onwards, I'm presenting you the results. On your left hand side, you can see the ethanolic leaf extract we prepared by crushing, and in your right hand side, this is C6 glioma cells in culture dishes where we captured from Olympus BX53 digital upright microscope under 100 magnification. So these are the scratch assay cell images. In A, we are having C6 glioma cells monolayer, and we made a scratch using a pipette tip, and we incubated these cells with 1000 microgram per milliliter ethanolic leaf extract treatment. Then with the time intervals of 6th and 24th hours, as you all can see the B and C images, the gap has been reduced, and finally it's not there anymore by image C, which means the cells have proliferated, migrated and closed the gap. So this proliferation and migration happens due to the effect of the plant extract we used. In D, we added the negative control, which is the distilled water, and the scratch remained almost same. Because of making the scratch using a pipette tip, some of the cells may remain in the gap, and 100% washing off was not possible. So as used, C6 glioma cells are already live and migratory cells. They do migration, so the general migration is there. So when we concluding the results, what we consider was the cells moving as groups but not as individual general migration. So in plates D, E and F, you may see the gap is getting a bit increasing, but just don't get confused. That is because in the same plate we just get 3 images and we averaged it when getting the results conclusive. And sorry, and in because of no, you may see the pictures are in different colors actually. That is in because of exposure levels are different. In fluorescence microscope, the bright field option is used. So depending on the visibility of the cells at that particular time, the exposure settings were changed. And there was an assumption throughout the scratch, the extract treatment is like uniform. So which means we got the averages, like we in one 35 millimeter culture dish, we had 3 images and 5 measurements we've taken per 1 image, and therefore 15 images are there, and we did 3 replicates. So which means finally we are having 45 measurements are averaged per 1 dish. Here are the TLC studies we did. So those chromatograms were developed in DCM, hexane, ethyl acetate system, having 3, 1.5 and 0.5 ratios. So A is crushed leaves in ethanol, B is leaf powder sonicated in ethanol, and C is crushed leaves in DCM, and D is leaf powder sonicated in DCM. So in A and C, chlorophyll-like plant pigments are prominent in the chromatograms, but B and D, those pigments are less, which means sonicated B and D extracts carry less plant pigment traces and more of other critical secondary metabolites. And by analyzing the RF values in developed chromatograms, leaf powder sonicated in DCM has 7 chemical constituents, while sonicated ethanol extracts had 4 chemical constituents. So this is the conclusion. The ethanolic extract having 1000 microgram per milliliter concentration had a positive effect on cell migration with 100% wound healing closure over a 24-hour incubation period. When considering the TLC chromatograms, they manifest sonicated leaf extracts had lesser chlorophyll traces than crushed extracts. And by analyzing the developed chromatograms, we could conclude leaf powder sonicated in DCM had more chemical compounds than the sonicated ethanolic extract. So in this study, as we did not conduct our scratch assay with crushed DCM extract, sonicated ethanolic and DCM extracts, that should be done as future work. In here we've used C6 glioma cells, which are internal migratory cells. For further reliable results to get the wound healing, we have to do the scratch assay with epithelial cells having migrational ability. For example, we can use Vero cells, which are green monkey kidney epithelial cells. Also, the column chromatography has to be continued to isolate the bioactive compounds in respective extracts individually. Then here are the references and acknowledgement for the personnels and the institutes who facilitate in this study. Thank you for hearing, and you may feel free to ask questions.
For that presentation, we have time for discussion, about 5 minutes, even though there was problem at the start. So you covered the time very well. So it's open to the house for any questions.
Yes, the cells were visible under our fluorescence microscope, in magnificent 100. Yeah, it was.
Check, right?
Yeah.
Thank you for that presentation. What is the plant extract you use? Naturally, in lay terms, what is it used for, that plant extract? Is it used as Ayurvedic medicine or?
No, it was used traditionally, like folks, they have used for cuts and wounds, like it is very indigenous one, but that had no any scientific evidence when we started this study.
So how could you find the plant? I mean, is it available?
Yeah, it is an invasive vine which we can find anywhere in our wetlands.
So you said that your next step is using vero cells and like that, in
Yeah.
using the different type of cells for
Vero cells, yes.
vero cells. But are you planning to expand for the animal studies further in future?
Yeah, if it is like going to be like more, positive for the vero cells and all, yeah, obviously we can go for that.
What about the toxicity of these extracts for the cells? Have you checked the toxicity?
No, not actually.
Not yet.
Yeah.
Before you introduce to the cell culture plate, in which medium you have dissolved that extract? Is it a cell culture medium or?
It's cell culture medium, yeah.
So then why you put distilled water in the as a control? You had to use the cell
Yeah,
culture
actually
medium.
the first one was aqueous, and then we thought of having cell culture medium as well. So like both means like worked as the same way.
Yeah, but you had to use the cell culture as a control.
Yeah. Yes. Not yet, madam.
So concentration 1000 milligram means microgram means whole overall a
Yeah.
mixture of compound.
Yes.
Even DCM extract will get more active compounds
Yeah.
like polyphenolic compound, phenolic or other some
Yeah.
compounds also. Okay.
Yeah,
So you
probably.
need to do the next step before
yeah.
implementing. No, the cell
Yes.
culture assays. I think best thing is to identify what is the really active compound and
Yeah, the thing is, madam, actually we did this for our undergraduate research studies. So
That's
yeah.
where, yeah, it's a very nice, interesting presentation,
Yeah.
actually. Thank you so much.
Can I just make a comment on your two solvents? Because although the DCM has more compounds,
Yes.
it doesn't necessarily mean that they're active.
Yeah.
So I was glad to see that you were thinking of doing that say with DCM, so you move in that. But there again you will have a problem in solubilization. Right?
Yeah.
Because you get a lot of more
Compounds.
non-polar compounds.
Yeah.
So that's the tricky thing. If you put surfactants, then you have to have a control
Yeah.
for surfactants, you know what I mean? But the other thing is, what's your strategy going to be about looking at active compounds? Are you going to, from what I understand, you're going to isolate compound from this and then test each compound, is it? The normal procedure we use would be to fractionate active
Compounds and then isolate.
fractionate the active extract into different fractions and then test.
Ah,
Regarding chlorophyll, it's not such an issue that you can get rid of it. There are enough techniques available for getting rid of
Okay.
chlorophyll. So that also you might want to consider before you plan the next stage. But it's interesting first results and
Yeah, okay.
congratulate you on
Yeah, thank you very
your
much, sir.
work.
Wound healing and cell migration in these cells.
Yeah, the thing is, sir, yeah, in wound healing, as like in epithelial wounds, like, so we just actually, that is what I said, for the future work, we actually do with the vero cells, which are epithelial cells. But the thing is, in wound healing, the epithelial cells are migrating towards each other. So, yeah, the wound healing, what do you call, the mechanism, yeah, there are several steps. So one of this was proliferation of the cells to much more towards healing of the wound or the cut. So cell migration means like, yeah, it's more.
You need to work with more standard
Yes.
cells for sketch wound
Yeah.
assay. But these are preliminary, I think. Okay, I think our time is up now.
Yeah.
So I thank Ms. Hewageegana for interesting presentation.
Yeah, my pleasure.
And call upon the next speaker who will talk about protein-ligand docking for atherosclerosis to uncover phytochemicals as therapeutic exercise. Paper is by M. Ifran, H. Mudalige and O. Perera and will be presented by Mr. Ifran.
To the head table, mighty judges and presenters, a very good morning to you all. I'm delighted to present about protein-ligand docking for atherosclerosis to uncover phytochemicals as therapeutic exercise. I'm also delighted to be a part of 15th International Research Conference at Surgeon John Kotalawala Defense University today on behalf of my co-authors Perera and Mudalige from School of Science, BMS. To introduce me, I'm Mohammed Ifran from School of Science, BMS. First of all, we are going to have a look on the intersection of atherosclerosis. The pathophysiology of atherosclerosis includes endothelial dysfunction, vascular inflammation, and the buildup of lipids, cholesterol, calcium, and cellular debris within the intima of the vessel. Atherosclerosis leads to stroke, which is the primary cause of adult disability in Sri Lanka. And Sri Lanka has one of the highest age standardized stroke mortality rates and social burden in Sri Lanka and as well globally. The symptoms of the atherosclerosis include chest pain or angina, pain in area that has a blocked artery, shortness of breath, and fatigue. If we move to the next, significance of this study, why we chose this atherosclerosis for our study. The complications and risk factor for many severe diseases such as stroke, heart attack, and angina inclusive of atherosclerosis. The current side effects of current drug treatment, such as antisense oligonucleotide against ApoB100, has injection site reactions, flu-like symptoms, and increased alanine aminotransferase levels, which leads to impaired liver diseases. As well, the MTP inhibitor has gastrointestinal disorders. To overcome this, the need to discover the new drug candidates are now on much concern. If we move to the objectives, to identify ligands as drug candidates, it has targeted. The background of the study includes atherosclerosis is the main risk factor for cardiovascular disease, which is the leading cause of mortality worldwide as for now. As it is an underlying condition of various serious diseases, the need to discover novel drug candidates is now on much concern, so that we perform this research. Targeting PCSK9 receptor, which binds to low-density lipoprotein receptor and accelerates the progression of atherosclerotic plaques, is a relatively new therapeutic strategy, apart from the currently available docking studies worldwide. These are the materials used for this research. It's a computational program, as hardware and websites and software were used with the i7 processor computer. If we move to the methodology, having said that, the receptor preparation was done by diluting water and heteroatoms to get rid of the binding pockets, misunderstandings. Repair missing atoms were followed for the receptor preparation by adding hydrogen and Coleman charges, and the Coleman charges was spread after that. This is done to prepare the receptor. Afterwards, the ligand preparation was done by converting .stf file to PDB-QT by creating a batch file to run the whole 100 ligands docking at a single file. The Autodoc Vina file was great. Configuration file is helpful to run this at a single run. Then the results were analyzed, as the slides number 7 shows. Having said that, we are on results now. The figure 1, a canadine, silybuline, hesperidine, narrogene blasis ball shows the best docking process of the ligands. As the table 1 shows, the binding affinity, the nilotinib is a control FDA-approved drug, has minus 11.7 kilocalorie per mole, and the silybuline had minus 10.7 kilocalorie per mole. And as the table 2 shows, analysis of phytochemicals analyzed in silybuline has accepted for Lipinski's rule of 5, and it has no blood-brain barrier permeability and low in gastrointestinal absorption also. The estimated solubility log value is moderate. Since the ligands, 100 ligands were docked, the best fit for the docking results are listed in this slide. These are the introduction and validation method for our procedure and methodology. Figure A shows the hydrogen bonds, hydrophobic interaction, pi-pi-alkyl bonds, and pi-sigma bonds as the van der Waals forces, conventional hydrogen bonds, carbon-hydrogen bond, halogen, pi-alkyl, and all are listed in this slide number 9. If we move to the interaction table, all the ligand has a common hydrogen bond in the amino acid register proline 331 and valine 460, proline 438, cysteine 358, proline 331 has identified as the most common amino acid residues with respect to hydrophobic interaction in the molecular study for PCSK9. We move to the discussion of these results. As per the previous studies for PCSK9 receptor, which has the PDB ID of 6U26, their studies showed the previous value was minus 9 kilocalorie per mole, which is lower than our study value minus 10.0 kilocalorie per mole, which shows the reliability of the current study done by us. And the second apomorphine showed minus 10.1, which is higher than the previous study done by Zainab et al. In 2021, minus 9 kilocalorie per mole. It shows, again, it proves the reliability of this study. And no hydrogen bonds were found for outperforming in the previous study done by Zainab et al. And in this study, we found hydrogen bond at arginine 357, which increases the ligand efficiency by hydrogen bond. And the water molecules and heteroatoms were deleted not to interrupt the binding site. Coleman charges calculated from electrostatic potential were added to 6U26 to calculate net atomic charges. The missing atoms are repaired to optimize and stabilize the physiological protein as per the human physiology. To identify the hydrogen bond interaction, the polar hydrogen atoms were added. VINA utilizes the gradient interaction algorithm with mechanical optimization. More the hydrogen bond and lower the binding energy increases ligand stability, as it showed in this slide number 12. We move to the conclusion. From FDA-approved drugs, nilotinib showed higher binding affinity, which proves it can be a potential drug candidate for atherosclerosis. The hydrogen bond and hydrophobic interactions are listed in the table 5. From phytochemical, silibnine showed higher binding affinity and Lipinski rules acceptance with moderate log S value. And the table 6 as well shows the hydrogen bond between TRP461 and SP360, which includes the 2 hydrogen bonds. We move next, the suggestions for future work. The network pharmacology and protein-protein interaction for this receptor can be done in future. In gene ontology and KEGG enrichment pathway analysis, particularly WNT pathway, can be followed and QSAR study. In the past, prediction also can be done for phytochemicals as well. The in vitro and in vivo experiments, such as atmospheric pressure, high pressure liquid chromatography, can be done. That concludes my presentation. These are the references, and my acknowledgement slide is shown in 16. Thank you for all listening to me.
Thank you for your presentation. It's now open for discussion. We have about 5 minutes available. Any questions or comments? Please. Mike.
Okay, yeah, it's fine. Okay, so thank you for the presentation. I'm just curious about the software that you use. So you have been using so many compounds. How long did you run this software to get this data?
It's approximately runs for too long with other softwares. As I did with the different techniques. It's a kind of new technique. I docked within an hour for 100 ligands.
100 ligands within an hour?
Yes.
Yeah, what is the storage you have?
My storage
Yeah,
for
memories.
laptop,
Yeah?
256 GB.
Oh, okay. So that small memory.
I
you
use
have-
only for this, for laptop.
only for this
Okay.
one, but still it's
Yeah,
fast
yeah.
one, right? Okay. Right, that's the question I had. It's a super fast one. Because you had done a lot of ligands, so I was curious how you did that one.
Sure.
Okay, thank you.
Okay, thank you. Since we can't afford the highly paid high-end software, I use with free software because the better software is like a Glide Schrodinger, $5,000 per annum. USA. Autodoc.
I have one question for you. And you said the last analytical method, you said API HPLC.
Yes.
So what is the API stands for? This is for my knowledge actually. I know what is HPLC, but I never seen API. What is that?
Atmospheric pressure and
Atmospheric pressure.
chromatography. Yeah.
Is API.
Atmospheric pressure interaction, I think.
Okay. What's actually? Do you know what it's actually doing?
No, because it's in future work. I found an article related to that.
Okay.
Yeah. Because it falls under wet lab. Since we are from green ethics and we didn't use wet lab procedures, we are into dry labs. So we didn't go for that wet lab procedures yet.
Okay. I
Yes.
was curious. That's why I asked.
Okay.
Thank you.
Okay, thank you.
What is the source of your data?
Data in CBI and PDB websites. And I found out some phytochemicals from different articles.
Yeah, it would have been better if you cited them.
I cited them because I have lots of phytochemicals in different articles for every phytochemical. So
Yeah, still you have to do it.
yes, I cited it at the end one. Yeah.
Okay, good.
Okay, thank you.
FDA approved drug is
Yeah.
used for what?
For nilotinib,
Yeah, you had a drug which had the highest binding. Because
Yeah.
yeah.
it's already approved. So we used
And it's
to
used for what? What clinical?
nilotinib. It's treat neurodegenerative diseases also.
Neurodegenerative.
Yes.
Not for
No,
atherosclerosis.
not approved for this disease.
Right.
Yes.
Yeah. Yeah. Okay.
Okay.
Any more questions? Okay. In the absence of any more questions, I thank the speaker for an
Okay,
interesting
thank you.
presentation and wish you luck with your future work. I call upon the third speaker today, which is on the green synthesis of silver nanoparticles using 5 varieties of leaf extract from Annona species, evaluation of antioxidant, photocatalytic properties and antibacterial properties. Paper is by D. Fernando and M Kandiah, presented by D. Fernando.
A very good morning to all of you. I'm Dinuka Fernando, and I'm proud to present my very first research topic, which is the green synthesis of silver nanoparticles using 5 varieties of leaf extracts from Annona species. I will be evaluating antioxidant, photocatalytic, and antibacterial properties during my research. These are the contents that I will be presenting to you today. We all have heard the term nanotechnology, but do we really know what it means? Nanotechnology is a developing field of research and development which involves in the design, fabrication, and characterization of different particles, materials, or systems. Nanometers is 1/1,000,000th of a meter. Nanoparticles are microscopic particles with at least one dimension in the scale between 1 to 100 nanometers. One of the key areas in nanotechnology is the synthesis of silver nanoparticles. And there are various methods to synthesize silver nanoparticles from. In my research, I used the biological synthesis method to synthesize silver nanoparticles. One of the key areas in nanotechnology is the green synthesis of silver nanoparticles, as green synthesis tends to be more eco-friendly and non-toxic in nature than chemical synthesis. It is more preferred. There are various applications of silver nanoparticles, such as being an antioxidant. It has wound healing properties and anti-inflammatory properties. Due to these factors, silver nanoparticles are more used in environmental degradation properties and in cosmetics and other various medical appliances. During the synthesis of silver nanoparticles, silver ion gets reduced to silver. Silver ions get reduced to silver by different phytochemicals present in the plant extract. Silver nanoparticles are more preferred than other metal nanoparticles such as copper, gold, or nickel, as they tend to have a large number of properties such as the surface plasmon resonance, high chemical stability, and less toxicity when you compare with the other nanoparticles. The word antioxidant has gained a large amount of public attention as they play a vital role in the human body, as they have the ability to protect the human body from free radicals. Antioxidants are also known as free radical scavengers, as they have the ability to donate an electron for the free radical, thus stabilizing it and preventing the rate of oxidation. Some of the benefits of antioxidants are given here, such as it aids in heart health, wellness, and in skin health. In the last few decades, due to rapid urbanization and population growth, it has caused a large amount of environmental pollution, as a large amount of toxic products, namely dyes such as methyl red, are being thrown out into waterways. Silver nanoparticles are found to have photocatalytic properties, which means silver nanoparticles have the ability to degrade dyes such as methyl red under visible light. By forming different radicals such as oxygen radicals or hydroxyl radicals, are capable to attack and degrade methyl red and form various byproducts. Another hazardous to human life is the ongoing strains of bacterial strains. Plant extracts are more researched and developed, as they provide an alternative natural strategy to kill bacteria, and as well silver nanoparticles have the ability to degrade and to kill different bacterium by causing oxidative stress. These are the objectives of my research: to evaluate phytochemicals from the 5 varieties of Annona cassia water leaf extracts, to synthesize silver nanoparticles from the 5 varieties, to evaluate antioxidant properties, to evaluate the nanoparticle shape and size, to evaluate the photocatalytic properties, and to evaluate the antibacterial activity. In this study, 5 varieties of Annona cassia samples was used. Annona are a common fruit that is very well known in Sri Lanka and throughout the world. Therefore, the leaves are widely not utilized in the commercial product. So, even though a number of researchers have shown that Annona leaves contains a large number of effective properties. The 5 varieties of leaf extracts was obtained, air-dried for 7 days, and 2 grams of sample was mixed with 50 milliliters of water, and it was heated up to 95 degrees Celsius for 20 minutes. The extraction was filtered, and it was used for to test out different assays. To synthesize silver nanoparticles, 1 milliliter of extract was mixed with 9 milliliters of 1 millimolar silver nitrate solution, and it was optimized accordingly, and the absorbances were measured. These are the different phytochemical testing methodologies that I used for this research, and these are the results that I obtained for each of them. Phytochemicals play a crucial role in the formation of silver nanoparticles, as they have the ability to synthesize silver nanoparticles, thus converting metal salts into metal nanoparticles. The synthesis of silver nanoparticle was optimized accordingly at 60 degrees Celsius and 90 degrees Celsius for 15, 30, 45, and 60 minutes. Even though there were no production of silver nanoparticles during this time, therefore it was optimized at room temperature for 24 hours. In the next slide, we have the first of all the observation. from a colorless solution to a dark red color solution after 24 hours, indicating the presence of silver nanoparticles. But a more reliable method to see the presence of silver nanoparticles is by the UV spectrophotometer. As you can see, a distinctive peak between 420 to 440 nanometers was observed for Annona murritata, Annona squamosa green, and Annona squamosa red species. These are the different antioxidant assays that are used to test for total phenolic content, total antioxidant content, and total flavonoid content. And these are the results that I obtained for each of them. The total phenolic content was determined through the FC reagent, and a higher total phenolic content was observed under silver nanoparticles than the water extract. The total antioxidant content was measured by the phosphomalidum assay. And here also higher concentration of total phenol antioxidant content was observed in silver nanoparticles than the water extract. The total flavonoid content was measured by the aluminum chloride iron calorimetric assay. Here also the higher concentration of silver nanoparticles was observed than the water extract. The nanoparticle shape and size was observed by the standing electron microscope, and it was in 40 nanometers in length and in spherical in shape. The photocatalytic activity of methyl red was observed under sunlight at two conditions, at sunlight and in the presence of sodium borohydride catalyst in the presence of two concentrations of 267 ppm and 4000 ppm. These are the results that I obtained for each of them, and a higher K value was observed at 4000 ppm under sunlight for methyl red. The rate was calculated by the pseudo first order rate kinetic law. In the other slide, we have the photocatalytic degradation of methyl red under sunlight in the presence of sodium borohydride catalyst, and here also a higher rate was observed. In comparison with the results that we obtained for sunlight, a higher rate was observed under sunlight in the presence of sodium borohydride. And as you can see in all four results, the peak gradually decreases over time, indicating the deprotonation of methyl red. Thus, it confirms that the diethyl and diethyl bonds are getting decomposed during the particular time. To check the antibacterial activity of the water extract and the silver nanoparticles, the petri tissues were labeled accordingly, and these are the results that I obtained for each of them. As you can see, a higher zone of inhibition was observed in the synthesized silver nanoparticles for both E. coli and Staphylococcus. In the next slide, in conclusion, out of 5 Annona leaves, 3 out of 5 samples produced silver nanoparticles at room temperature after 24 hours. Thus, it was known as the optimized temperature. The antioxidant assays were carried out for all 3 samples of silver nanoparticles and all 5 water extracts, and the nanoparticles showed a greater concentration of antioxidant content than the water extracts. The size and shape of Annona squamosa red silver nanoparticle was spherical in shape, and it was approximately 40 nanometers in size. The photocatalytic activity was carried out using methyl red for Annona squamosa red under sunlight and under sunlight in the presence of sodium borohydride catalyst, and a higher rate was observed. The antibacterial activity was analyzed by E. coli and S. aureus, and the nanoparticles showed a higher zone of inhibition than the water extract. Therefore, concluding, I can say that Annona squamosa leaves can be utilized as a therapeutic agent. These are the references that I used for this presentation today. I would like to acknowledge School of Science of Business Management School for giving me this opportunity to carry out this important project, and the Institute of Sri Lankan Institute of Nanotechnology for allowing us to use the scanning electron microscope. Thank you.
Thank you for that presentation. It's now, we can have a short time for discussion and questions if there are any.
It was a very nice presentation. Thank you for it. I have a very general question for you. I know many of the researchers are currently working on nanoparticles, synthesizing different kinds of nanoparticles. But have you ever thought of, but the thing is, these nanoparticles will end up in the environment. Have you ever thought of the destiny of these nanoparticles? Will that be a problem in the future, as we do have with the plastics right now?
Yes, so that's one of the key issues as well, even though it's it tends to be more non-toxic than other nanoparticles, but the research are quite low. And there was a study in US which basically talks about the disadvantages of silver nanoparticles, because it's not widely researched. So it do have some disadvantages, but in comparison, it's quite advantageous as well. So yeah, I'm not sure how to probably like further testing of silver nanoparticles. I guess at the end of the day, it does end up in the water.
I'm not talking about specifically on silver. I'm just talking about generally. Generally nanoparticles. Because when we were inventing plastics, I think scientists may not thought of that it would be a problem in the future.
Yeah.
But currently we are working on nanoparticles, but in future generations, this might be a problem.
Yeah.
Do you think that?
Yes.
Antimicrobial, the control you used to check was the aqueous extract. But properly speaking, there should have been also a control which used silver nanoparticles of the same size and dimensions, but prepared using a conventional chemical method. Because nanoparticles themselves have
antimicrobial properties.
So it's wrong to attribute what you see. Whereas if you had a control of nanoparticles which are synthesized without the plant extract, you may.
So yes, so what I have done is, I still didn't get the question, but so what I have done is, I compared it with the water extract without the silver nanoparticles and the synthesized silver nanoparticles. Yeah.
No, I'm just suggesting that if you're pursuing this to show that green synthesized nanoparticles have some advantage.
Yeah.
Say over antimicrobial activity.
Yeah.
Apart from environmental issues, then you need to have the correct controls to show that it is actually advantageous.
Thank you.
Other comments or questions? Okay, so
I just want to know, when you prepare the silver nanoparticles for the determination of the antioxidant activity, did you get the crude of silver nanoparticles or just you take the solution of the silver nanoparticles?
The solution. So it was filtered, and the solution was basically taken for both synthesized silver nanoparticles and the water extract.
I just asked for the antioxidant activity.
Yeah.
Did you take the crude, the solid form of silver nanoparticles?
Solid form of silver nanoparticles?
It means after you synthesize silver nanoparticles, you should centrifuge and get the nanoparticles, right? Did you do that procedure?
Yes.
Then you take that silver nanoparticles crude for the antioxidant activity.
Antioxidant activity as well.
Is it enough for 1 millimolar concentration silver nitrate solution for prepare the
Yes,
solid
so
form?
this was all for 1 millimolars, the synthesis of silver nanoparticle. Yes, it was for 1 milliliter of plant extract with 9 milliliters of 1 millimolar silver nitrate solution.
Okay, that's 1 to 9 ratio.
1 to 9 ratio.
Okay, thank you.
You're welcome, thank you.
And we can move on now to the next paper, which is characterizing surfactant effect of phytoremediation of carcinogenic anthracene by Iconia crassipes plant. Paper is by P.K. Vitanage, M.T.D. Perera, U.K. Samarasinghe, L.D.A.M. Arawala and P.M. Kolonna. paper will be presented by P.K. Vitanage.
Good morning, everyone. I'm Pramodi Vitanage. Today I'm going to present my research work, Characterizing Surfactant Effect on Phytoremediation of Carcinogenic Anthracene by Iconia crassips plant. This is a research work done by University of Kelaniya with Industrial Technology Institute. So what is anthracene? Anthracene is a toxic, low molecular weight, polyaromatic hydrocarbon which has 3 fused rings. Due to its low water solubility and low volatility, it's very resistant to degrade, so it pile up in soil and groundwater for years after year and make the environment polluted. So mainly, anthracene can be disposed into the environment via disposal of fossil fuels. Other than that, it can be disposed into environment via vehicle emission, sludge application, wastewater irrigation, and incomplete combustion. So, anthracene can harm the environment, animal health, and human health as well. Due to its low water solubility, it pollutes the environment. Mainly it pollutes the aquatic ecosystem. But it harms human health not only via aquatic ecosystem but also via food chain transmission. Since polyaromatic hydrocarbons are very lipid soluble, the human tissues can absorb the anthracene via the gastrointestinal tract and by the enzymes such as cytochrome P450 monooxygenases. It can be turned into epoxides and things like that and become genotoxic. And also it can form adducts and cause mutations which will end up in a cancer. So here we have used the phytoremediation technique to remove this anthracene from the contaminated area. So you can see some applications of phytoremediation in removal. So phytoremediation is a method of reducing the volume, mobility, and toxicity of contaminants in various environmental compartments such as soil, groundwater, and other contaminated mediums by employing plants and their associated rhizosphere. So here we have used water hyacinth, Iconia crassips, to use this phytoremediation technique because it has this ability to quickly reproduce, and also it can endure to a high hydrocarbon level even in toxic levels, and it can thrive in variety of freshwater settings, so that it has been ended up as an invasive plant in many countries. And also it's a floating water weed, and it has thick fibrous feather-like roots. It can absorb organics and convert them to other components, and also its nutrition requirement is very low, so this is a cost-effective method to remove anthracene. So the speciality of this research work is we have used not just phytoremediation technique, we have used surfactant enhanced phytoremediation. Because the polyaromatic hydrocarbons are very low water soluble, and due to low mass transfer and low bioavailability, it's quite hard to remove these kind of polyaromatic hydrocarbons via phytoremediation. So by using surfactants, we can just boost the efficacy of phytoremediation by using surfactants. So these are the 3 methods which explains how the surfactants increase the efficacy of phytoremediation. Those are first, dispersion of non-aqueous phase liquid organics. Second, micellar solubilization, which increases the apparent solubility of pollutants, and increase in contact area due to lower interfacial tension between the aqueous and non-aqueous phases. So we have used Triton X100 as the surfactant here. It's an anionic synthetic surfactant which has high desorption efficiency. It's less prone to sorbed into soil, and due to its low critical micellar concentration, we don't need to actually use high amount of surfactant here, and also it can form larger micelles due to less repulsion between the head groups. And our hypothesis of this research work is Triton X100 may increase absorption of anthracene by water hyacinth plant, and therefore can be an excellent candidate for surfactant enhanced phytoremediation. So the objectives of my research work are to qualitatively determine the presence of anthracene in the roots of the water hyacinth plants, to evaluate the possibility of using surfactants for phytoremediation by water hyacinth plants, to quantify the available anthracene in root samples to optimize anthracene absorption from the roots of water hyacinth plant. Here are some researches that have been done regarding the phytoremediation and surfactant usages. So in this particular article, they have shown that anthracene is the major component in contaminated soil samples. So this research work would be very useful to remove this major component from those contaminated soil samples. Let's move on to the methodology. First of all, I collected water hyacinth plants from a freshwater tank in Venapu area. Then all the plants were incubated for 1 week in fresh water prior to the experiment. And I prepared these 5 kinds of test solutions. Those are water crude oil anthracene, water crude oil surfactant, finally water crude oil surfactant spiked with anthracene. Then I grew the plants in these test solutions, and the plants were collected after 11 days and kept under 10 degrees until extraction. Then the plant roots were separated, cleaned, washed with a mild detergent and freeze dried, then grounded into uniform size powder, and the samples were dissolved in the extraction solution, which I used the dichloromethane acetone 1:1 mixture, and I used ultrasonication method for the extraction purpose. Then the solution was filtered and the volumes were reduced using rotary evaporator, further purified by doing a silica column chromatography to remove the unnecessary polar contaminants, and the fractions were collected which showed absorption at 225 nanometer. And then again the volumes were reduced using a vacuum rotary evaporator and dissolved in acetonitrile, and identification and quantification of anthracene in the root samples were carried out using the HPLC system and UV-DAD. There I used reverse phase chromatography, and we have used the C18 column as the stationary phase and water acetonitrile cyst gradient as the mobile phase. Let's move on to the results and discussion. Here you can see the average anthracene concentration in those different test solutions. In the graph you can see there is a huge difference in the plant water crude oil Triton X100 anthracene sample. It's actually 5.6 times more compared to the control sample. Here I have used a student t-test to analyze these results statistically. Here I have compared 4 types of test solution. In the first graph you can see I have compared water sample with the water crude oil treated sample. There was not any significant difference, maybe because I have not added surfactant here. And let's move on to the second graph. Here I have compared water crude oil treated sample with water crude oil Triton X100 treated sample. There we can see a significant difference between them, but surprisingly I got a result which I did not expect. Here I expected the anthracene concentration to be increased in the sample which I have used surfactant, but it has been reduced. This can be due to co-solubilization effect, because I have not spiked this sample with anthracene, so this sample only contains the crude oil. Crude oil is a mixed, it's kind of a complicated mixture of many polyaromatic hydrocarbons. So if the many polyaromatic hydrocarbons are present in a surfactant mixture, it can cause mutual inhibition or synergism. So here mutual inhibition has been occurred. And let's move on to the third graph. Here I have compared water crude oil treated sample with water crude oil anthracene treated sample. There was not any significant difference, maybe because I have not added surfactant here, but I have spiked anthracene here. So let's move on to the final graph. Here I have compared water crude oil anthracene treated sample with water crude oil anthracene Triton X100 treated sample. There you can see a huge difference in the surfactant treated sample. So this difference can be due to the addition of Triton X100. Let's move on to the conclusions. The water hyacinth plant can absorb anthracene in measurable quantities. Anthracene spiked crude oil did not increase the amount of anthracene absorbed by the plant in the absence of the surfactant. Triton X100 can enhance the absorption of anthracene from anthracene spiked crude oil sample. What are the future possibilities of this work? we can check on the distribution of anthracene in the water hyacinth plants. We have checked only on the root samples, so we can check it on leaves as well. And also we can use a chemical dispersant here instead of a surfactant. And also we can use symbiotic microorganisms along with the surfactant. And also we can check for other polyaromatic hydrocarbons as well, like naphthalene, phenanthrene and stuffs like that. And also we can find out a way to re-extract anthracene from the water hyacinth plants. And also we can characterize the mechanism, so we can understand fully about the mechanism here. So here are my references. And my acknowledgements goes to Dr. Punsiri Mahendra Kolane, Department of Chemistry, University of Kalaniya, Dr. LDM Arawala, Herbal Technology Section, Industrial Technology Institute, Sri Lanka, Mr. Kosala Samrasinghe and Mr. Menush Dilkar. Thank you for listening. That's the end of my presentation.
Two things I'm asking. The incubation for 1 week at the beginning, and then you went to low temperature extraction. The reasons, can you explain a bit?
Yeah.
Thank you.
So for the first question, we incubated the plants prior to the experiment for 1 week in fresh water to give the same conditions to the plants, because we collected plants from different areas in the same tank but in different areas to give them a same condition prior to the experiment. We incubated for 1 week. And for the second question, we maintained the extraction solution and the stuffs in 10 degrees to avoid any sample loss, because anthracene is somewhat, the volatility is quite high. So to save the sample, we did it.
After the extraction that you showed in the plant, so that's why I thought you kept the plant in 10 degrees. No, after the extraction you kept the extract in the 10 degrees.
The root samples as well.
Root samples as well?
Yeah.
Okay. Samples as well as the extraction you kept on?
Yeah, 10 degrees.
Okay. Right, thank you.
Can we go to the slides where you have a couple of plots where you were having different combinations of the substances and concentrations? I would
Slide
like
number 15.
Yeah, what is the concentration here? I mean, what? You don't tell, I mean, where do you find these concentrations, and what concentrations you are talking about here in your y-axis?
Anthracene concentration.
Yeah. Where? In water column?
In root samples.
In root samples.
In milligrams of anthracene per 1 kilogram of root sample.
Okay. Did you extract and measure them?
Yeah, I extracted anthracene from the root samples using the ultrasonication method.
Yeah, things should be clearer than this in your plots. You have, this is not clear enough. You have to mention the compound or whatever you are talking about there, right? So it's a very big drawback. Anyway, we have to be a little realistic here. Accepting the scientific importance and validity of the study and your effort, I have to say a few things as constraints in your approach. First, anthracene is not water soluble that much, right? So you try to work with the water column, not even the sediments, even if you remove all the possible amounts of anthracene from water column using your technology, still there should be plenty of anthracenes and other polycyclic aromatic hydrocarbons in the sediments. So you are not going to do any substantial good with your technology for that water body. That is one thing. The second, you want to clean the environment, but yet you add something totally synthetic to the environment. That is your surfactant. I'm not sure whether that is acceptable at this era of science and technology, right? So please think about these things.
Yeah, let me add some comment on that question. If you can remember. Can you give me slide number 5?
The other thing, let me add. The plant of your study is an invasive plant, right? If you add an invasive plant and Triton X100 into a water body in order to remediate the water body with some polycyclic aromatic hydrocarbons, I'm not sure whether what would be the net effect from the point of view of the environment.
Yeah, true.
Right. So please, we have to be realistic in doing things. But as I said at the first, in my first few sentences, I accept the scientific importance and the validity of the approach and the study and your data. But the era where we played around with the environment is gone now. We have to be absolutely sure that we are not adding any further questions in that area.
Yeah, true. I accept that, sir. Actually, we are not planning to expose water hyacinth plants directly onto the water bodies. We are kind of planning to do this as an application like this. We are planning to take water, if let's say anthracene is contaminated in soil or sediment samples, then we are planning to dissolve it in water and make this kind of tanks and put the plant and surfactants like this. So we don't adding plants or surfactants continuously. We are we are continuously using the same plants and same amount of concentration. As you can see, there are different tanks. One by one it passes to one by one. We are just not releasing it into the environment.
So
and
then,
we are
you need to mention that in your presentation as well as in any material which you will be writing of.
Sure. Thank you.
Any further discussion we have to do later, but thank you for those comments. I also have some points regarding the realism of the total objectives, not only in this paper, but in other papers. Is there a question? No. No. Okay, so then thank you for that presentation.
Thank you.
And we move on now to the next one, which is again on nanoparticles. The evaluation of in vitro antioxidant potential of multifaceted phytogenic zinc oxide nanoparticles synthesized from peel waste extracts of Annona's varieties. Paper will be presented by AMS Athanayaka. Authors are AMS Athanayaka, TG Thiripuranathar and S. Ekanayaka.
Good morning, all of you. Thank you very much for introducing me. My presentation today is evaluation of in vitro antioxidant potential of multi-phased phytogenic zinc oxide nanoparticles synthesized from peel waste extract of Annona's varieties. This is a part of my MPhil research work. I'm Samudrika Attanayaka, a postgraduate student of Institute of Chemistry, Ceylon. This is the overview of my presentation that I hope to discuss during next 12 minutes. As we know, nowadays, use of the green nanotechnology for the synthesis of nanomaterials is an innovative technique for the nanomaterial synthesis, and it also contributes for the environmental sustainability. Biosynthesis of nanomaterials using plant extract is eco-friendly, and also plant-mediated nanomaterial synthesis is has a good role. It has a significant role in the field of nanotechnology because this plant extract contain so many phytochemicals. Actually, it is a rich of biocompound profile. So these bioactive components act as reducing, capping, and stabilizing agents for the synthesis of nanomaterials. Accordingly, synthesis of zinc oxide nanoparticles from agro waste, especially in here peel waste extract, is a very good application for the nanomaterial synthesis. According to our objective was green synthesis of eco-friendly zinc oxide nanoparticles utilizing peel waste of commonly consumed pineapple varieties and comparative analysis of their antioxidant potentials. Initially, we collected our raw materials from cultivated land, mursi and cube peels, that pineapple varieties. In order to synthesis of nanomaterials, we optimized the conditions, especially the concentration of the iron precursor solution, the zinc acetate. Here, the concentration of iron precursor solutions and plant extract to iron precursor solution addition ratios and irradiation methods like UV, microwave, and solar addition methods and incubation times. According to that, the optimized condition for the concentration of iron precursor solution was 100 millimolar concentration, and plant extract and iron precursor solution was 1:1. Then the optimal irradiation method was microwave irradiation method. Under the 24 hours incubation time gave the optimum yield of the zinc oxide nanoparticles. After that, the synthesized nanomaterials characterized under UV visible spectrophotometer, XRD, FTIR, scanning and transition electron microscope analysis. For the determination of the antioxidant activity, we carried out DPPH and ferric reducing antioxidant power assays. This is the diagrammatic form of our methodology. Initially, we collected raw materials, mursi and cube peels. Then those peels are dried for get the powdered form of peels, and we prepared the plant extract. Then, to determination of the antioxidant activity, we followed standard procedures for the DPPH and ferric reducing antioxidant power assays. Let's move on to the research and discussion part. Initially, synthesis of the zinc oxide nanoparticles, we determined through UV visible spectrophotometer the surface plasma resonance peaks. The characteristic surface plasma resonance peak for the mursi peel mediated zinc oxide nanoparticles under microwave irradiation method, it was 359 nanometer, and cube peel mediated extract zinc oxide nanoparticles gave the peak at 356 nanometer. Then, under the FTIR analysis, the FTIR patterns for the peel-mediated zinc oxide nanoparticles and respective peel samples. Here, the peaks for the biosynthesized zinc oxide nanoparticles, there are more peaks than their respective plant material. The reason for that is, after the synthesis of these nanoparticles, their surfaces contain bioactive compounds which act as capping agents. So therefore, there are more peaks in biosynthesized zinc oxide nanoparticle samples than their plant material. Then formation of hexagonal phase of zinc oxide and formation of tetrahedral coordinated zinc. In here, the FTIR patterns, the frequency range between 710 to 900 shows the formation of these hexagonal peaks, stretching peaks of zinc oxide nanoparticles formation, and 500 to 600 frequency range shows the stretching mode of zinc oxygen bond vibration patterns. When we move on to the XRD analysis, when we move on to the SEM and TEM analysis, the morphology of synthesized biogenic zinc oxide nanoparticles using Mursi peel mediated, it shows spherical shaped nanoparticles, and Q peel mediated nanoparticles shows needle shaped or needle shaped nanoparticles. And transition electron microscope using Mursi peel mediated zinc oxide nanoparticles, it get 10.23 nanometer size, and zinc oxide nanoparticles prepared from Q peels, it was 5.44 nanometers. When we move on to the XRD analysis, it's indicated that the formation of hexagonal phase of zinc oxide nanoparticles with respective to their 2 theta peaks and crystalline planes. Here, according to shear equation, we get the crystalline sizes of the prepared nanoparticles. Zinc oxide nanoparticles under Mursi peel mediated, it shows 14.7 nanometer, and the zinc oxide nanoparticles prepared from Q peel, the crystalline size was 14.5 nanometer. When we move on to the antioxidant properties, antioxidant activities of these nanoparticles, we compared the prepared biogenic zinc oxide nanoparticles with respective plant materials and also iron precursor solution as well as the reference standards. Here the DPPH radical scavenging activity of biogenic zinc oxide nanoparticles, antioxidant activity of these zinc oxide nanoparticles are higher than their respective plant material as well as zinc acetate iron precursor solution. When we move on to the ferric reducing antioxidant power assay, this also indicated that the considerable antioxidant potential of phytogenic zinc oxide nanoparticles. Finally, the conclusion here, in our study, we indicated that the successful formation of zinc oxide nanoparticles using aqueous peel waste extract of Q and Mursi peels, pineapple varieties, and the formation of pure hexagonal form of zinc oxide. In here, Q peel mediated zinc oxide nanoparticles gave the smaller size, crystalline size than the Mursi peel mediated zinc oxide nanoparticles. And when we consider about the morphology of these nanoparticles, even pineapple plant, if there is two varieties, the Q and Mursi, Q and Mursi gave the different types of shapes of these nanoparticles. Here the smaller size nanoparticles gave the Q peel mediated zinc oxide nanoparticle with needle shape, and Mursi peel mediated nanoparticles gave the spherical shape nanoparticles. And finally, the antioxidant activity, when considered about the plant material, their respective zinc oxide nanoparticles gave the highest antioxidant potential. In here, according to the smaller size of the Q peel mediated zinc oxide nanoparticles, it gave the higher antioxidant activity than the Mursi peel mediated zinc oxide nanoparticles. These are the references of we followed throughout our study. And finally, I would like to convey my gratitude for my supervisors, Dr. T. Gobika and Professor Sagarika Ekanayaka, for their valuable guidance throughout this study. And also, I would like to convey my gratitude for the College of Chemical Sciences for their financial supports and laboratory facilities. And thank you very much.
Thank you for your presentation. Open for discussion.
Thank you for the presentation. I'm asking this question out of curiosity again because your purpose is to prepare zinc oxide nanoparticles. So actually we can very easily prepare this using salt gel or hydrothermal. Why you specifically go for this
Green
field
synthesis.
extract? Yeah.
Yeah, green synthesis method normally now, according to when we consider about the sustainability now today, so we mainly focus for the synthesis of nanoparticles using green method. According to that, actually we focus for the synthesis of nanomaterials using agro waste. That is the main thing because when we use fruit like fruits or food, it's better to use agro waste to synthesis of nanomaterials. So that's why we focus for the synthesis of nanomaterials using green method.
So when you got this zinc oxide sizes, you controlled it and got it on purpose, or it automatically happened? I mean the needle shape and so on.
Yeah.
That is, you controlled any condition there to get the needle shape, or your output was little like that?
These nanoparticles, we optimize the condition for the formation of nanoparticles. As I said earlier, it's mean according to the radiation methods, the iron precursor solution concentrations and ratio of adenine iron precursor solution to plant material. Because of these conditions, their particle sizes, shapes, morphology also change. So that's why we got the different size, different shapes of these nanoparticles. Q-peel mediated zinc oxide nanoparticles gave the needle shapes, and the Mursi-peel mediated zinc oxide nanoparticles gave the spherical shapes, like that way. So that's why.
So that's fine, because I was curious. The zinc oxide you prepared, it's only zinc oxide, and this antioxidant is just coming because of the shape. That's my curiosity. Because
maybe.
no other thing is there. It's only because of the shape. The difference is there.
It's maybe like that way, madam. When we use these nanoparticles, it's prepared from green methods. These plant material also have several antioxidant activities as well. Sometimes these bioactivities contained in plant materials also can affect for the antioxidant activity as well. So it boosts the activity. So that is the one of the advantage of green synthesis method of preparation of the zinc oxide. So according to your question, the antioxidant activity, the shapes, sizes also may affect for the antioxidant activity as well. Additionally, the plant bioactive compounds also affect.
Okay, thank you.
Thank you.
Thank you for your presentation. I have just one general question about the green synthesis of nanoparticles. So I have seen previous presentation as well, there is some kind of a leaf extract to synthesize green nanoparticles. So my question was to you is, do you know what is the actual mechanism?
Green
to form
method. Still there is no any actual mechanism, but according to several characterization, FTIR and sometimes same analysis, XRD analysis, we could get several ideas. If the plant contains several phytochemicals like flavonoids and peptides, something like that, those are used for the synthesis of these nanoparticles. Because of when we consider about the FTIR analysis, the corresponding peaks for the OH bond, which contain in flavonoids, phenols, and also NH bond in peptides, something like that bonds are disappeared in the nanoparticle FTIR patterns. These peaks are disappeared. According to that, we can get an idea these phytochemicals are responsible for the nanoparticle formation. Other than that, there are no any exact mechanism for the synthesis of these nanomaterials.
Okay, thank you.
Thank you.
Mechanism, how is the zinc acetate converted to zinc oxide?
Here.
Is there anything known about that?
Yeah. We add sodium hydroxide initially.
You add sodium hydroxide?
So
you convert to hydroxide?
Yeah.
Okay, that's fine.
Other than that, without adding sodium hydroxide, we also can prepare zinc oxide.
You can get zinc oxide?
Very low yield.
Yeah, that may be some other thing. All right. Because you're not, it's a chemical conversion there. Right? Yeah, okay. Any other queries or comments? Okay, so thank you for your
Thank you, sir.
interesting presentation, and we move on now to the last presentation for this session, which is again on antioxidant activity, in vitro antioxidant and anti-inflammatory activity of leaf and stem bark of selected Mangifera species in Sri Lanka. Paper is by S. Hassan, P. Ranasinghe and A.I. Kuruppu, presented by S. Hassan. Is he not here? The paper will be presented online. Person is not here personally for some reason.
Good day to all. My name is Sadana Hassan, and I'll be presenting our research in vitro antioxidant and anti-inflammatory activity of leaf and stem bark of selected Mangifera species in Sri Lanka. In Sri Lanka, the two most common species of Mangifera is Mangifera indica and Mangifera zelenica, and the most common among them is Mangifera indica, which is mostly known for being rich in health-enhancing compounds, as they are known to be experimented for its phytochemical, pharmacological, and ethnomedicinal properties. So in this research, we carried it out to determine the antioxidant and anti-inflammatory effects via bioassays for ethanol extracts of varieties we selected, which are Columban, Willard, Velai Columban, their leaves and stem bark. The mango fruit of the three varieties are shown in figure 1. Firstly, we obtained the plant samples and made a herbarium sheet and confirmed whether the samples we are using are the varieties we selected. It was then air dried, and we did an ethanol extraction, as it can be seen in the image. Then we did relevant assays. I'll be going through the results one by one. The results we have obtained, we have presented in terms of which out of the three varieties have the highest phenolic, flavonoid, antioxidant or anti-inflammatory activity. These are the herbarium sheets which we prepared to know the validity of the samples. In table 1, the percentage yield of the extracts can be seen, and we used a standard labeling from number 1 to 6 throughout the procedure for the specification of the samples. Firstly, we have done the TPC assay, or the total phenolic content, which works on the principle that the formation of blue complex compounds, they form due to the reaction between the phenolic compounds and the folding C reagents. The wavelength was 765 nm for the assay, and as it can be seen in figure 6, we used gallic acid as a standard, and we found the absorbance values for the samples 1 to 6. For the TPC assay, results were calculated as gallic acid equivalents. In TPC, the highest value was recorded for Columban bark, which was of 3.33, and the lowest was for Velai Columban bark. In the total flavonoid content, or TFC, the main principle was the ammonium chloride forms acid stable complexes with the C4 keto group and either the C3 or C5 hydroxyl group of the flavones and flavonols. Here we used quercetin as a standard, and at 405 nm the absorbances were found. And for the TPC, the results were found as quercetin equivalent. Here the Velai Columban leaf and Willard leaves had the highest TPC. The DPPH assay is based on the measurement of scavenging capacity of antioxidants. In figure 9, the percentage of scavenging activity of extracts compared to ascorbic acid with the standard can be seen. 520 NM was the wavelength used here and ascorbic acid as a positive control. When the percentage inhibitions were found and compared at different concentrations, Willard bark had the highest rate of percentage inhibitions and Columban leaf had the lowest value noted. As seen in figure 5, the IC50 values were calculated indicating the concentration of antioxidants which is required to lower the initial DPPH concentration by 50%. It is assumed that the samples with low IC50 values has a higher antioxidant activity, and here Velacolumban bark has the lowest value, which is of 0.0029. The oxygen radical absorbance capacity or ORAC assay result relies on the free radical damage on the fluorescent probe, most commonly fluorescein, caused by an oxidizing reagent resulting in the loss of fluorescent intensity over the time, relating the resultant damage to the amount of oxidants present. The excitation wavelength was 494 nm for this assay, and the emission wavelength was 535 nm. Ascorbic acid was used as a positive control. This assay was done following the DPPH result. It was noted that Willard bark and Willard leaves had the highest percentage inhibition, so ORAC was done on those extracts to validate the DPPH results. When the ORAC values were calculated, the highest value was of Willard bark. So it could be said that the DPPH results do collaborate with the ORAC results we have obtained. The HPRC or human red blood cell membrane stabilization assay was also done where the erythrocyte membrane is comparable to the lysosomal membrane, and its stabilization implies that the extracts may as well stabilize the lysosomal membrane. On that note, ibuprofen was used as a standard here, which is also a non-steroidal anti-inflammatory drug, and the absorbances were taken under the wavelength of 560 nm. When percentage inhibitions were calculated at different concentrations, Willard bark and Columban leaves had the highest values. As seen in the graph, here when compared to the standard ibuprofen, highest percentage inhibition was seen in Velacolumban bark, Columban leaf, and Velacolumban leaf. The lowest IC50 value was calculated in Willard bark, which was of 0.0010. In plant samples, TLC or thin layer chromatography provides an indication of a number of compounds that can be present in the extract through a color separation. In figure 14 to 15, after the color separation to mark the spots, the chromatography paper was visualized under a UV cabinet. Among the tested extracts, Columban bark had shown the presence of multiple compounds which can be phytochemicals after the RFL was calculated. There are similar experiments done, but the results are different, and it could be said that the conditions used for this experiment differs from the others, so there is a change in the overall result of ours. In case of DPPH, based on an experiment carried by Ito et al., it was said that different stages of mango can differ in their antioxidant activity, as they have found that pruned mangoes of a different species, Mangifera, even has a scavenging activity of young dark reddish brown leaf extracts higher than the others, and the overall lowest DPPH value was the lowest ORAC also in all dark green leaf extracts. In case of ORAC assay results, while trying to compare the results, we went across an experiment of Soji et al., where they have concluded that different drying techniques gives different results, and since we used air drying techniques, our results can be different from the results obtained through different drying techniques. In case of HRBCSA, a similar experiment to this research was carried by Koganison et al. On the peel, seed and pulp extracts of columban, Villard and Velar columban. As per their results, the columban samples had the highest anti-inflammatory activity and Villard had the lowest, which to some extent agrees with our results. Columban leaf had shown two prominent spots from our TLC, where the other varieties had showed one spot each when the RF value was calculated. The solvent system was, which was used for our TLC was dichloromethane and 30% methanol in 1:3 ratio. So if a different solvent system was used and if left for a different time duration, the results obtained might be different. So as per the findings of this research, we can conclude that the tested three Mangifera varieties do contain antioxidant and anti-inflammatory properties at varied levels, and this data can be useful when perhaps considering to make a food supplement or a pharmacological agents in the future. These are the references used to make this presentation. I would like to thank my supervisor, Dr. Anshula Kurupo, for her guidance and advice throughout the research. Also, Dr. Rani Singh and staff at Industrial Technology Institute of Sri Lanka for providing the laboratory facilities and guidance necessary. And a heartfelt thanks to the staff and my colleagues at KDU for the help and support given throughout the laboratory works. Thank you.
Thank you for the presentation, Sasan. It's open for discussion now. For any comments, I assume that she is online and can hear us. Can you hear us?
Yeah. Yes, I can.
Yeah. Okay. So, are there any comments or questions? Okay. So, in the absence of any questions, we thank Ms. Hassan for her presentation. And we now come to the end of the presentations. I think there is some time available in general for discussion. If anybody has any questions regarding the papers that have been presented up to now in this section, are there any from the audience that would like to comment? Otherwise, I think what is remaining is for me to just make some summing up comments. First of all, I'd like to thank the presenters for their efforts. They have done careful laboratory work and come and presented their results here. But thinking a little ahead, particularly regarding the overall objectives of scientific research, is also important, I think, particularly for those who are going on to become researchers, to think about doing significant scientific work and not merely adding more empirical data into the system. And there are two areas currently where unfortunately this is taking place. One is in terms of antioxidant activity. Now, antioxidant activity, which was a very big thing, say 20, 30 years ago, has now become very controversial. And in fact, the US, which was thinking about ORAC values for foodstuffs, removed that classification and have deleted their database from 2010 onwards, because there is no evidence that antioxidants we take with our foods have any bearing directly on any particular health problem. And even with regard to medical applications, there is no definite evidence. This is not to say that antioxidant activity in our body is not important, it's very important, but the question of direct ingestion of antioxidant material and its effect on the body is questioned now. So, in fact, I think we need to move away now from merely reporting antioxidant activities of various plants, because it's of not much theoretical significance or of practical significance. If you are to work on antioxidants, I think you need to get down into more detail into molecular mechanisms and whether it's targeted to the particular cells that you want to, etc., which require very much more sophisticated experimentation, which may not be available to most of us here. That's the sad thing. And I think supervisors who are choosing research projects for their students with the very limited facilities here sometimes have no choice but to give things. But I think it's important to realize that while you are learning techniques, that also think about the significance. I think the same issue cropped up in the environmental issues as to really how practical what is being suggested is. But whatever it is, the important thing is that the work is done properly, and I think we saw that here. The laboratory work has been done carefully, the results are results we can rely on. I'm only talking about what happens after. So that may be something for those of you all who are going on in the future to become scientists. I think when you're planning projects, according to the facilities, you have to pick something where you add something, knowledge of significance to. For example, now when you take, this is other area is this creation of nanoparticles using various biological things. Now that itself is simply due to the antioxidative properties of the plant materials. If you take silver nanoparticles, silver is very easily reduced, so you know they're correlated. But if you are going to go further, you need to really see what are the chemicals that are responsible, what are the conditions which will give you different sizes of nanoparticles. And then we talk about encapping and absorption of material without ever talking about what particular material is being absorbed. And you say, and again the use of control, as I said, almost all of these papers we have presented today, as well as the hundreds, thousands of papers published in the literature, when you want to compare the activity of a nanoparticle synthesized using a plant extract, and then what do you compare it with? I think you have to compare it with a similar particle which has been synthesized using normal chemical methods to show that your biogenic thing actually made a difference, and then try to find out what the difference is. So you need to get into the next level of research now, because both these things are now very well documented in the literature. If you say, I made nano silver by using the leaf of this plant, you can do it with the leaf of the next plant also. Regarding antioxidative activity, some journals say that they will not report papers, they will not publish papers which are merely reporting antioxidant activities. So you need to think a little bit about that. But beyond that, I would like to congratulate all the speakers, because they presented work which I think has been done very carefully and with reliable results, and that's the most important thing. Is only to plan experiments to have more significance. So with that, I thank everybody here, the presenters, the audience, for their participation in the discussion, and bring this session to a close. Thank you. You wish to make a comment? Oh, she's making it.
On behalf of the organizing committee, we apologize for the technical issues that arose during the session due to ISP network errors. It was indeed a very insightful session. We are grateful for our chairperson, Emeritus Professor A M Abeysekera, for his contribution to make this event a success. Now I cordially invite the Assistant Director of KDU Care to present the token of appreciation to our chairperson, Emeritus Professor A M Abeysekera. Thank you, sir. This officially ends the technical session 2 of Basic and Applied Science track. We'll meet again with the technical session 3 after a short lunch break at 1 p.m. Thank you.